All right, bear with me. I'm doing this by myself. Hello? All right, so hello, my name is Charlie Shellhorn, and I am here to talk to you about grapevine resistance towards downy and powdery mildews. All right, so a little bit of background about me. I work at a winery. I work at Luce Winery. And infection of grapevines at Luce equals lower varietals for work, which means less money for me. So the more resistance we can apply, that will increase production, which will increase money for me. Also, my roommate, he's the assistant winemaker, and his brother-in-law, my good friend, is the head winemaker at Luce. They're also coming out with their own label, their brand label, called Accent Wines. So you could say wine is pretty much my life, or my life is pretty much wrapped around wine. So we're going to start with a little bit of background on downy mildew. All right, it's caused by a heterophthalic diploid called P. viticola. All right, which is an oomycete. All right. After 5 to 7 days of infection, you're going to start to see an appearance of on white grape leaves, you're going to see more of an appearance of a blotchy yellowness. All right. And for the red grape leaves, you're going to see more of a mossy appearance that some people do confuse with the powdery mildew. Okay. Now for the grape clusters themselves, which we pick for harvest, the grape clusters are going to turn brown. All right. After they're going to wilter, they're going to shrivel, and they're going to die rapidly. So this is going to be a little bit of background on what it looks like. So you can see here's the brown, the wiltering. They do die very rapidly right there. You can see he is starting to turn. All right, and this is going to be the grape varietal, or this is going to be a white grape varietal here because you have the yellow coloration happening here, not the mossy look. All right, that red grape varietals give you. All right, and so a little bit about powdery mildew. It's caused by a fungus called U. necatar. All right. This happens in most high-value grapes. They're very susceptible to the powdery mildew. Some varietals that you're probably familiar with are going to be Chardonnays, Rieslings, all right? And then you have your blended grapes. It's like your Concords, your Trollingers, and your Lembergers, all right? A few causes for downy mildew. It needs a lot of moisture and precipitation. That also plays a role in downy mildew as well. It affects all green leaf or all green tissue on grapevines with a whitish gray coloring, powdery coating. All right, on the leaves of the grapes, on leaves or grapes. Okay, and it's caused by the fungus mycelium. All right, once precipitation occurs, then the mycelium and the corneta on the surface then soon has a white lesion effect. All right, so. As you can tell, you do have that powdery look. It literally looks like you dipped it into flour, okay? After this, you're going to get the wiltering, the browning, and you're going to get death. You can see kind of back here in the fuzzies that you have the browning and the death, and it happens very rapidly. All right, so a little bit about the experimental background. For breeding purposes, this is why you want to apply molecular markers. It saves time, and it saves money down the road. So they crossed the VRH. They crossed VRH with regions. This is gonna be your male. This is gonna be your female. The region species is gonna carry quantitative resistances towards downy and powdery mildew. Okay, and this varietal was found in Germany. The VRH is gonna be the mother varietal, and this is gonna be a hybrid. Okay. It's going to be crossed with your muscadine, which is a common grape down here. You make very, very sweet wines. It's called the redneck grape. And then that's crossed with your Malaga Sealing No. 1, which is a V. vinifera varietal. There's about 5 to 10,000 varietals out there of V. vinifera. And so what they did is after they crossed this, they back crossed it again with 4 other species of the V. vinifera. Now we're going to be into methods. So after crossing your region with your VRH, your F1 progeny came out to be 119 individuals. All right, and like I said, they were derived from your VRH and your regions. They grew all of these varietals in a greenhouse with 1 genotype per plant, or 1 plant per genotype. Plants were experimented at the age of 4 years. Okay. So they're still young grape, or they're still young vines, but they're matured enough to have grapes. Okay. So a little bit into the VRH and the Regent. So the VRH carries two highly resistant genes towards downy and powdery mildew. You ever run one gene which is resistant to powdery mildew, and it's linked to the CAPS marker GLP12. And then you have your RPV1 gene, which is related to the resistance of downy mildew. All right. And for your father figure or your father varietal, you're going to have your Regent. All right. And this species is just a complex hybrid of red grape varietals. And it has a very complex resistance with a lot of alleles that are connected towards resistance to downy and powdery mildews. All right, so for the methods of downy mildew, we're going to break it into methods of downy mildew and then methods of powdery mildew. You have a phenotypic screening with artificial inoculation tests. All right, so what they did is they cut one leaf off per plant. All right, and they harmonized it, being the seventh leaf from the shoot top of a well-developed cane of each plant. After that, they took 4 leaf discs per leaf and placed into a plastic box with a wet filter paper. They used 20 microliters of spore suspension with a concentration of 50 spores per milliliter for artificial inoculation of downy mildew. All right, and so after 1 week, screening took place of the downy mildew, and this experiment was done twice, just for reassurance factors, for consistency. So what you're looking at here now is the downy mildew screening, and this also applies for the powdery mildew screening. They use a generalized OIV descriptor ratings. It's what grapevine cultivars use universally for rate of infection, for class of infection, not class of resistance, but class of infection. So it goes 1 through 9, all right, 1 being no infection, 3 being slight infection, all the way up to class 9, which is more than 50% of the several bunches per plant are infected. So bunches are the clusters of grapes that you see. That means half of those clusters on one plant itself are infected. All right, so methods for powdery mildew. They allow for a more natural appearance or natural process of artificial inoculation. They still artificially inoculated the leaves with the powdery mildew, but they just left it up to the plant itself. They didn't cut off leaves, they just left it up to the plant itself to watch for resistance or infection. And this went on for 2 years. Like I said, the screening's the same. They use the OIV descriptor ratings, still reading for levels of infection, not levels of resistance. You still have your scale 1 through 9, 1 being no infection, 9 being severe infection, and 50% of the clusters on a plant, individual plant. So for downy and powdery mildew methods, they did a DNA isolation on young leaves, and how they did this is they used a DN Easy Plant Mini Kit or the Thomas ETAL. I couldn't find much information on that, so for the next slide I have a little brief description of the DNA Easy Plant Mini Kit just to show you how it went down. They found related markers for both the RUN1 gene and the RVP1 gene, which is related to the mother varietal, the VRH. Okay. So this is your simplified explanation. Using a centrifuge, before you're gonna grind, lyse, and precipitate the leaves. After that, your young leaves. Now you're going to centrifuge it in a QIA shredder. All right. You're going to add ethanol to bind to the DNA of interest. After that, you're going to wash it. Then you're going to elute your sample, your DNA sample. All right. So after that, they did a QTL for a genetic analysis. All right. And QTL is a quantitative trait loci or loci. And they did that for downy mildew, and they also did that for powdery mildew. They found 3 SSR markers for each, okay? Within the QTL for downy mildew, they found linkages on group 18, and then for powdery mildew, they found linkages on group 15. They also found scar markers related to powdery mildew, or the scar marker score A7, 6, or 760 on that QTL strand. Or loci, pardon me. loci. All right, so for the results for powdery mildew, how I'm going to do this is we have 5 figures. I'm going to break it down, each figure. I'm going to give you a little description before the figures so you can kind of wrap your head around it. But the left is going to, you have 2 graphs on this first figure. The left is going to be a total progeny percentage related to infection. And the right is going to be, it's going to be divided into the run1 gene, which is related to resistance of powdery mildew. Either you have they're going to have the absence of the run1 gene or the presence of the run1 gene, okay? All right, so right here, you have your scale, the OIV scale, 1 no infection, 9 severe infection, all right? Here's your percentage of genotypes. So you're going to have 48% of the genotypes that show no infection of powdery mildew, all right, which is, that's what we're going to focus on for this figure. And then if we come over here, like I said, you have the presence or absence of the run1 gene, which comes from the mother varietal, the VRH. And as you can see here, we have a 100% success rate in class of infection 1, which is no infection whatsoever. So that shows a strong correlation of resistance to powdery mildew for the run gene. All right. For the second figure, we're going to be looking at the mother and father figures. All right, so we're going to be looking at the VRH, presence or absence of the run1 gene, and we're going to be looking at the region markers, which are about 3 or 4 markers, and it's going to be the presence or absence of those markers. And then it's just going to show you a correlation of rate of infection with the presence or absence of both of those. Okay, so we have four graphs here in this figure. Okay, the ones on the left are going to be the absence of your run1 gene, and the ones on the right are going to be the presence of your run1 gene, as you can see up here. The ones on top are going to be the absence of all of your region markers, which are right here, so you don't have your region markers present. And down here on the bottom portion, you're going to have the presence of all your region markers. So it's divided into mother, father, in a sense. So with everything absent, you have the absence of the run1 gene, the absence of all markers. You can see that the level of infection, you have nothing in here that shows no infection. All right, and for the second, and for graph B, you're going to have the absence of the run1 gene from the VRH mother, but the presence of the markers related to regions. All right, and so you can see a little bit of a shift coming over here into slight infection to no infection. All right, and now we're going to come up here. Now you're going to have the presence of the run1 gene, which is related to the VRH, and then you're going to have the absence of all markers that are related to the region varietal. Okay, we have a 100% success rate of no infection with just the run1 gene, which is a strong correlation, like figure 1 showed you, to resistance to powdery mildew. And then this is showing us still the presence of the run1 gene, and then the presence of all related markers of the region. There is no difference, it's just more resistance. You still have a 100% success rate for no infection. Now we're going to move on to downy mildew results, okay? It's going to be the same 2 figures just for downy mildew, all right? Like I said, figure 3A is going to show you class of infection to total progeny, and then class B is going to show you absence or presence of the RPV1 related marker. And the RPV1 related marker, if you remember, is also found in your mother varietal of the VRH, not the regent. All right, so like I said, we're going to have just the total progeny over here, and then we're going to split it up between the RPV1 absence or presence. So right here we have 36% in class 1 showing no infection of the genotypes, and then we have 3, with the class 3 we have 27%. Okay, over here now we split the presence or absence of this marker. For the presence of the marker, you have a 40% success rate of no infection. With the absence of the marker, you still have a very high percentage of 33% showing the no infection of downy mildew. In class 3, you still have a high number of individuals showing slight infection, and you still have a high number of individuals with the absence of the RPV1 related marker with a slight to little infection. All right, so figure 4 is going to be just like the second figure I showed you. Figure 8 is going to show you that 74% were class 7 or above, so that shows you that we have severe infection. That's going to be the absence of both father-related marker alleles and mother-related genes, the RVP related genes. Figure B is going to show you the presence of all resistant-related markers in regions, okay? And you're going to see that we have a 96% of it laid in classes 1 and 3, okay? And then I'll explain figure 4C, figure 4D to you here in a second. All right, so this is going to be the absence of all mother and father related genes. All right, once again, you have the absence of your mother related genes, the presence of your mother related genes, the absence of your father related genes, and the presence of your father related genes. So this is the absence of both. You're going to have 74% of these individuals highly infected in classes 7 or 9. Now when we move down here, this is just going to be the presence of your father related genes. This is going to be about 96% showing classes 1 to 3. That's individual grapes that are infected to no grapes that are infected. And we have more no grapes class 1 infected than we do in class 3. And then we're going to move on to C. This is going to be the absence again of all your father-related alleles or markered alleles. And then this is going to be the presence of your mother-related RVP1 gene, all right, or markers. It shifted significantly from 1 to 3. You're showing slight infection to almost medium infection with the absence of father-related genes, showing a huge correlation between resistance of the markers found in the region varietal. And then when you combine the two, the RVP1 related markers from the mother and the markered alleles from the region varietal, you're going to have a 100% success rate of no infection. Showing you that it's the best varietal out there is going to be combining these two together for the least resistance towards downy mildew. All right, so the last figure I'm going to show you is going to be a pyramid. The first, they're going to show you a total progeny, and then what they're going to do is they're going to artificially inoculate it with spores of downy mildew. They're going to take out of those 119 progeny, the ones that are not affected or that show resistance to downy mildew, they're then going to artificially inoculate them again with powdery mildew. All right, and out of that number that show resistance to downy and powdery mildew, they're going to do genetic analysis tests showing marker related alleles, and then for downy mildew, and then they're going to repeat that process again, the genetic analysis for powdery mildew, and this is what they get. So, like I said, you have 119 individuals, okay? This is going to be your total offspring of your VHR in your regions, okay? After that, like I said, they're going to artificially inoculate it with downy mildew, all 119, and you have 44 individuals that show no infection of downy mildew. After that, they're going to artificially inoculate all 44 individuals with powdery mildew spores, okay? And this shows that 20 individuals have no powdery mildew infection. After that, they're going to do the genetic analysis that I went through for powdery mildew, showing that there's 9 individuals that hold those marker-related alleles, all right? And then one more time, they're going to do a genetic analysis for the marker-related alleles for downy mildew, and it comes up with 4 individuals that show resistance to downy and powdery mildew, total resistance to downy and powdery mildew. So for my conclusion, powdery mildew, the RUN1 gene is highly resistant, or the RUN1 gene GLP1-12 is highly resistant and linked to powdery mildew resistance. Okay, and then these are the markers from the father varietal that show resistance. You have 19% of them that show no infection. You have 30% of them that show slight infection, class 3. All right. On to Downy mildew, the RPV1 gene, which is in the mother varietal, is linked to Downy mildew resistance, but it is not nearly as strong as the resistance coming from the father varietal for Downy mildew. Region definitely has a higher resistance, has higher resistance related alleles to Downy mildew. So it shows you right here that we have 71% is in class 1, no infection, and then we have 25% in class 3, which is slight infection. So that tells us 96% of the progeny that have these related alleles from a regent father are going to be pretty much Downy mildew free or show slight infection. Okay. All right, so for my future studies, I would like to find varietals with molecular markers that are highly resistant to Downy mildew. Reason is because Downy mildew is the #1 pathogen in North America. Also, grapevines in Georgia, not many varietals can grow here in Georgia. We have red clay, which is hard for good grapes to grow down here. So I would like to find varietals that we could breed down here that would be resistant to Downy, or Downy mildew and or powdery mildew. Also with this, this would save us time. This would save us money buying fungicides and tractors to spray the fungicides. Also, these fungicides can't hurt humans. And what people want to do when they go to wineries is they want to walk the wineries. They want to walk within the grapes. They want to look at the grapes. They want to pick the grapes. Well, you can't do that because of the fungicides. You can't go in there. You can only go in there in certain times of the years. So this would eliminate that factor. This would eliminate the fungicides and allow possibly for more revenue for allowing people to walk through the grapes more frequently. So these are my references. All right. And that was my presentation. Thank you.